Hi, I'm here to show you the process of preservative decanting and evaporation performed after receipt of field samples. Before we get started, it is always important to practice good laboratory practices, which at this stage in eDNA processing requires sterilization of the equipment and workspace, changing gloves at the completion of each step in the process and if contamination is ever suspected, and clean gloves should be used if keyboards, pens, notebooks, etc. are handled. Additionally, different lab coats are worn in each lab area where open tubes are processed to further prevent cross-contamination. Upon receipt of the samples, each tube should be inspected and gently swirled if any debris from the pellet broke loose. Each tube should then be placed in the centrifuge, making sure to keep the centrifuge properly balanced and centrifuged at max speed for 5 minutes. You'll notice that our max speed is 4200 RPMs. These speeds may differ from machine to machine. While the samples are being centrifuged, you should clean your workbench with DNA away and set up a clean tray and a collection container for decanting. Once the centrifuge has completely stopped, place each tube into a clean tube rack labeled with the appropriate case number and sample range. Make sure to keep the samples together and set on the clean workbench. Remove caps and carefully decant each tube to ensure the pellet remains at the bottom of each tube. Placing the tubes back into the tube rack and placing the caps in numerical order of removal on the clean tray. Absorbent matting is used on the trays to keep spills to a minimum and also helps keep the caps from sliding around on the metal tray. The samples are now ready to be dried. Place the open tubes in the laminar flow hood and turn on the airflow until all traces of ethanol or isopropanol smell are gone. This process typically takes 24 hours from when the last tubes are placed in the drying hood and should never take more than 48 hours. Along with the samples, a hood control is needed. Place a clean 50 milliliter centrifuge tube in a clean tube rack labeled with the case number and sample range that makes up the control. Label the cap too and place on the tray with the rest of the samples. There should be at least one hood control for each case, and if an individual case is spread between multiple hoods, there must be one hood control per hood to ensure sample integrity throughout subsequent processes. Bottom line, if you put any samples in a drying hood, they must be accompanied by a hood control tube. It is very important to ensure complete evaporation. Both ethanol and isopropanol are PCR inhibitors, but also equally important to not let the dried samples sit too long at room temperature. With that being said, it is important to have your last sample of the week drying before noon on Thursday to ensure samples do not sit over the weekend. Additionally, if samples are drying at different speeds, place dried samples in the -80 freezer while waiting for the rest to dry. One additional tool that can be used to achieve completely dry samples are metallic drying beads in a lab armor bead bath set at 55 degrees Celsius. The use of the bead bath should be limited to extreme cases where samples are not drying within the 24-hour period that is preferred. This method is only to be used to avoid prolonged exposure in the drying hood. While you wait on the samples to dry, you can label the tray with the caps on it to the corresponding samples and place them in the tray rack. You can then clean the workbench area by using liberal amounts of DNA away and wiping the surface with clean paper towels. Once the tubes are dried, always assume there was a possibility for contamination, so clean your workbench again with DNA away and clean paper towels. Retrieve the tray with the corresponding caps and place them on the workbench. Then place the tubes in the hood control alongside it. Spray the emptied interior of the laminar flow hood with DNA away and wipe clean the interior and all touch surfaces. Then close the doors, turn the blower off, and turn the light switch to UV for 10 minutes. You can then carefully match the caps with the corresponding tubes, making sure to secure the caps without cross-threading. After all tubes have had their caps replaced, you can dispose of any tray liners or other non-reusable items. Bring the sample tubes along with the hood control tubes to the -80 freezers. Place the samples in a freezer and put which samples are on which shelf of the freezer on the dry erase board and initial and date. Then fill out the logbook. Always make sure to use a pen with blue ink to fill out logbooks. From there, return to the lab area and fill out the benchtop logbook. Then proceed to the computer and update the case log. You can then clean up the workbench and tray with DNA away. Put the tray back on its rack and turn off the UV light on the hood. The samples are now able to be processed as time permits. Just remember, always look to the co-op if you have any questions about how to proceed.